The next property of the proteins is to have its own isoelectric point. Now I have written the notes, you can read from there. Now please remember there is a pH called as isoelectric point. So we are talking about a certain pH at which a protein is electrically neutral. Now why is it neutral? Because it carries equal number of negative charges and positive charges. So there is a pH which is called as isoelectric point at which a protein is electrically neutral, and the reason is equal number of positive and negative charges. Proteins have the tendency to precipitate at this pH. So a pH at which the charges are going to be equal in their amino acids, now the protein is going to precipitate, and that point is called as isoelectric point. Now remember that we all know about milk, that milk when we start preparing yogurt or curd, we would have seen that the milk protein starts coagulating down. That's why curd is better than the milk, because it is partially digested type of milk. So the digestive problem people easily are suggested by doctors to go for yogurt or curd instead of milk. Reason is same, because its pH is going to precipitate. pH will be 4.7 pH. So at this pH the milk is going to coagulate, or you can say protein of the milk, that is called as the casein. Milk protein is called as casein protein. So this protein is going to coagulate, and this will cause the partial breakdown of protein, and this is good for those who have digestive problem. So let's understand that the milk forms curd at a pH which is 4.7, and most of the cytoplasmic proteins have an isoelectric point at 6 pH. So maximum proteins present inside the cell will have an isoelectric point at how much pH? 6 pH. So that's why at this point proteins are either basic or otherwise they are acidic in nature. So proteins can be neutral, acidic or basic. Now the reasons I have already mentioned here. Protein would be called as the neutral protein when it has equal number of negative charge containing amino acids and equal number of the positively charged, like if it is 8 here, it will be 8 here. So equal number of negative amino acids and positive amino acids. If you remember, we have done already this thing that amino acids may be neutral or may carry negative charge or positive charge. Please revise the notes, you will find it there. Why negative charge is there or positive charge is there. Now acidic one. Acidic proteins will be positively charged, and the reason is but they carry more number of positive charged amino acid compared to the negative charged amino acid. And basic are those which will carry a negative charge, and the reason is they carry more number of negatively charged amino acids compared to the positively charged amino acid. Now the next property you can learn about the protein is the solubility. So some proteins are insoluble in water. Remember we have learned already that amino acids are soluble, reason they are small biomolecules. When we talk about proteins, proteins grow longer in their chain. So some proteins are insoluble in water, some will form colloidal solutions with water, and remember the larger the size of the molecule of protein, colloidal nature would be present. So more long the chain is, more colloidal nature will be there. Otherwise you will find some of them are soluble also. Now the next is the denaturation. What is denaturation? And it is of two types, temporary denaturation and the permanent one. So what is denaturation? We'll be learning about the structure of protein on the next page. So there is a concept which is called as maintaining shape. So the moment a protein loses its shape or loses its activity center, then you will start calling it as a denatured protein. It can be for DNA also, RNA also. We will be learning in this chapter. So denaturation is but when the three-dimensional structure of a protein is lost. It is called that it is undergone denaturation. Now denaturation can be temporary or it can be permanent. Protein denaturation occur when bonds are broken. See, I have already told that this bond is required for stability. So there are various bonds which will stabilize the structure of protein. But sometimes what happens, with special conditions or treatments, it is possible these bonds may break. Once these bonds are broken, the structure of the protein is going to be lost, and we call that loss of the structure as denaturation. So whatever is the activity, if it is an enzyme, if it is a hormone, then all those activities are also lost. But remember, denaturation can be a temporary one or it can be a permanent one. So now why exactly denaturation, when exactly denaturation occurs? So try to understand that there are amino acids which are arranged in a specific sequence, and then because of loss of the conformation, the denatured protein is going to lose its function also. And the reason behind denaturation is ultraviolet radiation. It can be heavy metal, it can be a strong acid treatment or strong alkali treatment, or heat. Heat means infrared radiation, a detergent, organic solvent, alcohol. There are many reasons when the protein come in contact with any of these factors, it is possible it may break the bonds here and there, and the structure of the protein is lost. And once the structure is lost, protein becomes non-functional. It can be a temporary one, means if there is not a major change, when there is no major break of the bonds and all, and if it is possible to make the bonds again, it is called as temporary. But sometimes what happens, the treatment is so strong that there is a permanent bond breakage and no more renaturation is possible. What is the opposite of denaturation? Renaturation. So if it is a temporary denaturation, renaturation is possible by providing it good conditions again. And if it is a permanent loss, then renaturation is never possible.